Hi, my name is Christian Nansen. I am very proud to contribute to the 47th Congress of the Colombian Entomological Society, and I'm particularly interested because of this year's conference has a theme of frontiers in entomology, which I find very, very intriguing. The presentation that I'm about to share with you is divided into three separate sections. And even though they are very different in their content, they share some denominators that I think are very important for how we should look at ourselves, look at our discipline, look at our responsibility to the students that we engage with on a daily basis, and also how we see our role in society overall. So, and if you're interested in more information about what I do, here's the link to my webpage. But let's get started. I will be talking specifically about optical sensing, remote sensing, also referred to as. So this is use of imaging technology in identification, classification, and potentially sorting of insects. I will be talking about a project we have in which we are developing a phone app in use to optimize and optimize use of pesticides and how to optimize pesticide applications to provide a tool for quality control. And finally, I'll get into a completely different project or topic, which is mass rearing of insects and use of insects for bioconversion of agricultural waste. So from the get-go, you can see I'm combining three things that have very little in common, but I hope as we go along that you will appreciate that there are some denominators here that tie this together and sort of enable me to convey a broader message about frontiers in entomology and the importance of technology in particular, how we as professors should look at and embrace technology and multidisciplinary research in our teaching and education. I think it's very important to think back a little bit and remember that for the last several hundred years, it's been sort of the responsibility and the privilege of scientists to be driving the technological development, that it has been scientists who have basically led to all the discoveries that we have in society. What I think is happening here these last 30 years is that things are kind of being turned on its head in a way, where technology is now driving, say, scientific development. That many of us are now in the research we do and the teaching we are engaged in is in so many ways affected by technologies that are not only helping us but also dictating and showing dictating ways to move forward and how we should proceed. And I think that's driven by of course, the technology becoming far more accessible, affordable, much easier to use, and very robust. So I think technology, whether we like it or not, I think is in so many important ways affecting our society, but also how we should view entomology as a discipline. And then I want to highlight the two things that I have sort of put in red boxes here, and that is, I think a growing number of us are looking at research publications as one way of producing scientific and educational outputs, but just one of many. And some of us are maybe going a couple of steps further and thinking, well, maybe it shouldn't be the end product. Maybe we should go beyond that. And maybe society demands more from us than, quote unquote, just produce a scientific article. Maybe we have even bigger responsibilities ahead of us. And maybe we can be to even more value and be even more relevant to students if we are willing to go steps further. If we want to talk about and embrace entrepreneurship in developing smartphone apps and looking at technologies and see how we can turn it into patents and other things. So really looking ahead and providing solutions to many of the dire challenges that we as a society are facing. When we say we're entomologists, well, what are we actually doing on a daily basis? How many of us are actually collecting insects or dealing with insects on a daily basis? Well, many of us are not at all, right? So we do modeling, we do statistics, we do molecular biology, genetics, we do chemistry, all these different things. We may even be using different imaging technologies at different scales, work with drones. We may even do hardware and some robotics and programming as part of this. In essence, and coming from Denmark, I want to use this analogy with Lego blocks. We are combining sort of disciplines or research areas, and we as Lego blocks, and we apply them to insects in different ways. And I really think we can and we should, when we look forward and when we look at the frontiers of entomology, we have to look more broadly and start adopting more of these Lego blocks as part of what we teach and what we research, and therefore also how we can stay relevant to students and attract them to the discipline of entomology by combining it with these other Lego blocks. I will argue that as we look ahead in entomology, just like in many other academic disciplines, software development, hardware development, and especially entrepreneurship are going to be aspects that we should include as part of our curriculum, as part of our research. We see that funding bodies are focusing more and more on how the impact, what we do relates to society, and we have to provide metrics of that. Well, how about we also develop entrepreneurs? How if we as professors also were in charge of startup companies and patents and development of solutions? I think that's a very intriguing and inspiring way of looking at entomology as a discipline and looking ahead. I'm going to be talking about optical sensing, and we're going to be talking about optical sensing on a benchtop scale, spatial scale. But before we get into that, I just want to make a couple of important claims. First of all, I want to argue that I think optical sensing, the use of imaging technology, depending on the areas, it's called machine vision, remote sensing. It's all based on the same thing. We use different sensor technology to remotely characterize different objects at different spatial scales. And I will argue that is becoming the new molecular area. Just like in the 90s, or the availability of molecular tools started completely changing how we are performing research, I think imaging technology is doing the same thing. So I want to make that claim. And the second claim I want to make with this is with the visuals here is that we are essentially, from the imaging technology, from the sensors we're using, we are acquiring very high value, high spectral resolution imaging data. So we get these reflectance profiles that can inform us about objects in different ways. And this is not black magic. There is a lot of physics. There's a lot of math behind this. So even though it may in some ways sound and appear to be somewhat what's going on here, it's a little bit like the initial in the first years of the molecular technology. I think we are still working out some of the details, but there's some fundamental physics and phenomena behind it, which I will talk briefly about, to hopefully convince you that this is not black magic, that this is worthwhile, and there's a reason why the military, pharmaceutical industry, the medical area, the mining industries, all these, and the agricultural sectors, of course, all are embracing imaging technologies at different spatial scales using this data in all sorts of ways. And this is really an opportunity for us as entomologists to jump on that wagon and start integrating it into what we do and how we inspire our students. Optical sensing is the use of imaging technology, and typically we can mount that imaging technology on different, either in a stationary way, on an optical, on a benchtop scale, and that's what I'm going to be talking about today. But fundamentally, it's the same technology we use when we deploy it with a drone, with a rover, with a satellite. We have many options at our disposal here, and fundamentally it's the same thing. There are, of course, very important differences in addition to the spatial scale, and one of them being that if we are outside and we are dependent on solar light, then that the composition and the intensity of sunlight changes and the atmosphere changes and all that. So there's some calibration that needs to be included into this. But fundamentally, the use of the technology is the same at all these spatial scales. But I'm going to be talking about it specifically when we're in the lab and we have a camera that is mounted to acquire data at high spatial resolution, so from insects themselves, and at high spectral resolution where we have lots of spectral bands associated with each pixel. Probably some of the main reasons why optical sensing is being adopted more and more widely is because this technology allows for what's called high throughput. It's very fast. You can process and acquire a lot of data in a short amount of time. It's therefore cost effective. You can replace mechanisms and procedures that are typically very subjective and based on visual inspection or others. You can automate it, you can make it repeatable, you can also make it quantitative. And then lastly, and that's really important, it is non-destructive, meaning we can look at objects without damaging them. We don't have to take tissue samples and crush them up and then therefore lose them. We can actually just have a sensor, look at them, examine them by collecting their reflectance or transmission data, and then use that data to make some characterization of that particular object. So it's used very widely in quality control, classification, sorting, and again at multiple spatial scales, right? So if this is part of conservation efforts to find out what is the health status of a vegetation where we have insect communities that we are trying to preserve, and as I will talk about here, we can also look at the insects themselves and actually take images of them and use the reflectance to say something about them, and they can be both dead and alive. A key aspect in applications of optical sensing technologies is that we are often looking for the outliers. We are looking for that needle in a haystack. We are either scouting through or processing through a sample of insects, and we're trying to find that unique insect, that capra beetle, or that insect that is unique to us. Or we are flying over a crop field and we are looking for that hot spot where the spider mites seem to have established in a strawberry field. So it's the combination of fast, automated, repeatable, quantitative, all those things allows us to, in a fast way, process and find the outliers that we are typically looking for. That's what this technology really lends itself well to. An aspect of optical sensing that is extremely important for the use in entomological research and teaching is that even though we think of we acquire surface reflectance, we point the imaging device onto the surface of something, just like in this case the hand we project the infrared thermometer onto the hand. What we're really doing is we are assessing what's going on inside the hand with that thermometer. So what is important is that surface reflectance data also actually includes information about the tissue layers underneath the surface. So over on this curve here, or on this figure, you can see that particularly bands in the 800 and 900 nanometer range, so out in the near infrared, will actually penetrate several millimeters into lung tissue. So this is widely used in the medical field in their research. We as entomologists can use this because it means that we can, when we have sensors that collect acquired data in those wavelengths, we can actually quote unquote look into the insects. It's not like X-ray where we can actually outline the individual organs and whatnot, but we can get some information from the internal status of the insect, and that is critically important when we want to look at differences, say, between males and females, even if the morphological features are very, very difficult to distinguish. Maybe they have different fat body content, maybe they vary in some other ways that can be big or small, but at least it explains why it's possible, based on optical sensing data, to make inferences about these objects and how they vary. As an example of a proof of this penetration that we are looking inside objects, I'm going to tell a little story here. So I've already mentioned that I am from Denmark and we like Lego. Another thing we like is Hans Christian Andersen and the fairy tales that he wrote. And one of them, of course, is about princesses and how you find out whether a woman is a princess. And so this is the story about the princess and the pea, right? So we all know now that if you want to know if somebody is a princess, you have to offer her to sleep somewhere and then put a pea underneath a pile of mattresses. And if she wakes up and is all sore in her back, well, there you go, you have a princess. So inspired by this story, I published this experiment, actually, where I had 3 colored dots. These are my peas here, a blue and a green and a red, printed on a piece of paper. And then I put mattresses on top. Those, in this particular case, it was just sheets of paper. And so the question I wanted to find out was, how many pieces of paper would I need to stack on top of my colored dots for the imaging data that I collected right on top of the dots, for those data no longer to show effect of the dots, of the peas, so to speak. And the graph to the right actually shows the results. So if the pea was blue, it only took me about 5 sheets of paper for that dots to disappear. But if it was green and even red, I would need as many as 30 sheets of paper. And what you can see here is that the highest number of sheets of paper were needed in the 800 to 900 nanometers, which is exactly the data I was showing you prior on the other slide from lung tissue, where the medical field are seeing the same thing. So whether we're looking at lung tissue or whether we're looking at lignified paper, we're seeing the same thing, that the penetration of these spectral bands is highest in that 800 to 950 nanometers. And that just confirms that we can look into insects and learn something about their internal structure, their internal composition. And that is precisely the foundation for what we are trying to accomplish when we use optical sensing in classification of insects, or when we look at whether they, for instance, in this particular that I'll show here in a little bit, is whether they are vectors of plant pathogens. There's a couple of important take-home messages from this study that was inspired by Hans Christian Andersen. And first of all, so I demonstrated through a series of experimental studies where some of them included insects, but some of it actually didn't. But they were critically important for me to afterwards make the claim and continue to make the claim that we can, quote unquote, look inside insects. And the second claim I want to, or take home message I want to bring up here is that because I was using a technology and sort of a cutting edge technology, I had to venture outside the conventional entomology zone. I had to sort of accept that I needed to go outside the comfort zone of where I am as an entomologist. So that was clearly needed, and I think we are seeing that more and more across entomological disciplines, that people are now, because of what they do, have to go in different directions and venture out and work with people, that economists or with programmers or engineers or others, in ways that we haven't seen much of in the past, but we will see more and more of. And finally, this whole study was clearly driven by the technology. I didn't think of this initially as something I needed to do, but in order for me to use the technology properly after this, I needed to inform myself. I had to do the experiments, and then I decided to write a paper about penetration and scattering, about optical phenomena as an entomologist, right? So you can imagine how much deep water I was on here, but I got away with it. It took a while to get it published, but I did. But that's clearly, to me, an example of how the technology is now driving research rather than the other way around, the way it was in the past. A couple of years ago, we reviewed the technology of optical sensing, remote sensing, and its use in entomology. So in this review article from 2016, you can find lots of examples, lots of studies in which this technology has been used for species identification, for all sorts of physiological studies, and also in ways, for instance, to assess products with and without insects. So in the stored grain entomology area, with and without parasitism, they're just and symbiont biology. So lots of examples of how this technology has been applied and will be applied more and more in the future in entomology, in specific studies of insects, as well as on other spatial scales when we go out and look in agricultural areas and areas targeted for conservation and whatnot. In this study of beet leafhoppers, the context, of course, is that beet leafhoppers are the only known vectors of beet curly-top virus to tomato and other crops in California. And growers are restricted in their ability to use insecticides to control beet leafhoppers. First of all, because lots of products are not labeled, and many of them don't work particularly well. And thirdly, the biology of leafhoppers makes control of them very complicated, because they live long and they migrate in over an extended period. So when exactly are you supposed to spray? Now, the beet leafhoppers themselves are not particularly damaging to the crop. It's when they carry the beet curly-top virus that is transmitted during feeding and probing. So just because you have leafhoppers in your tomato field doesn't necessarily mean that you have a problem. And that is really the big issue here. So could we come up with a technology that would allow us to sample leafhoppers and then in a fast, repeatable way have a diagnostic tool that could be used to determine, are we having a population that is carrying the beet curly-top virus that are virulliferous, or are these mainly non-virulliferous individuals? So it's also about not just presence, absence of the virus. It would be also very important if you sampled, say, 50 or 100 leafhoppers. Could you then analyze them individually and then find out what is the proportion of individuals? If you did the PCR analysis or some other sample, you would get a positive or a negative, but you wouldn't know how many of the individuals were actually carrying it. So if we could do this with imaging, there would be huge advantages to this. So that's why we started having some colonies of leafhoppers that we then established on different crops with and without the virus, and then we followed them over time. And what you can see to the right, and there are different aspects of this paper that I'm not going to talk about today, but what you can see is if we have positive controls, we could detect that with 100%. And we could then also, as the beet leafhoppers acquired it, as they acquired more and more, there was also a progressive increase in their infection rate, which is what we would expect. A couple of the values are a little bit off, and that is to be expected. That it is not nearly as accurate as PCR is. Of course, it's not. But on the other hand, it's super fast, and it would be much cheaper in the long run, and you could get individual data. So instead of having to wait a long time and only have one data point from a group of individuals, you could then have individualized images from individual data points. So that gives you much higher resolution. Even though some of them may not be accurate, you're getting a much broader picture that at least could complement some PCR. Or you could argue that if you have a lot of samples, that maybe you could use it as a first type of screening. In other words, we're coming up with a technology here that could really help inform growers on when to spray, to avoid spraying when it's not necessary, and really use these pesticides when they absolutely have virulliferous populations threatening their specialty crop. What I think is really important to emphasize in this particular case is that we have a very important problem in front of us, a problem that growers and the tomato industry, the specialty crop industry is facing, and of course we do the research that then educates students and all that, and we get the study published, but along the way we are clearly thinking beyond the publication, that the publication is one step, but the real goal is down the road to develop a diagnostic tool that can then be integrated into diagnostic labs, or in other ways create job opportunities and professional training for our students. So it's very important in the way we approach this problem, not only to look at this, not simply just because we got a publication about this that we can then okay, check that box and now we're done. No, that's when the job actually really starts, is now we have the credentials, now we have the knowledge base that allows us to go to really work with the problem, which is how do we take it to the next level? How do we develop something that growers can use? How can we get it out in the real world? We're now going to jump into the second section of this lecture and talk more about insecticide spray applications. And here the context is also, just like when we talked about beet leafhoppers, is also related to an agricultural issue. In this particular case, the context is that pesticides are being sprayed excessively. And there are multiple reasons for that, and we all suffer the consequences of that. Growers are not interested in it; they are wasting money, and there are lots of environmental concerns and concerns about pesticide residue on the food we eat, consume, and on our backyards being sprayed and contaminated in all sorts of ways, and water bodies. So, of course, everybody's on the same page here. We all know that pesticides are may be necessary in some cropping systems, but we want to use them judiciously, meaning we want to only use them when and where they are needed. And in some cases, weather conditions and or the operational settings, the nozzles, the spray volume, how fast the tractor is moving, all those factors can negatively affect the outcome, and therefore lead to a spray failure and or a contamination of neighboring environments. So that's something we all want to avoid. Now, how can we develop the tools? So one thing to help farmers avoid these situations, because one thing is, as a scientist or as a consumer or as a parent, to be concerned about these things happening. But it's really a responsibility of us as researchers to find solutions to this. It's not enough that we stand up and get concerned and tell growers not to use as many pesticides and use them the right way. Well, we can't do that without providing them the tools they need. So this is something that, even though I'm not a spray engineer, even though, there are many things that I'm not, I think this is very, very important because I am related to applied agriculture, and I'm very interested in sustainable food production. So this is a project that we took on. How can we develop a tool that growers can use to make smarter decisions on when and where to spray their pesticides, if that is needed? Just to create a little bit of context here, I think it's important to start by looking at, why is spray coverage actually important? Why is it so important that this product that's being applied is ending up in the crop and not somewhere else? And we have performed some very, very simple experiments which sort of underscore the whole idea here. So to the left, you can see 3 cabbage leaves, and what we did, we put the 3 into a cage, but before that, one of them had been dipped in a insecticide formulation at the labeled rate that would have gone out into a field, and this was with diamondback moths. The one in the middle with the yellow sticky note had been dipped. Only half of the leaf had been dipped into the solution, and the red one, the entire leaf was dipped. So we knew that we had 0, 50, and 100% coverage. And then we released female moths into these cages and then saw where they laid their eggs. And we did experiments with 2 different strains of diamondback moths. And you can see in the graph, especially the one in the middle, when we used gamma cyhalothrin, that both strains strongly preferred to lay eggs on the non-treated leaf. In other words, they're able to assess in some way that this leaf has been treated, and therefore they try to avoid it. Okay, it was a little bit different with spinodoram, which was a chemical they weren't quite used to, but the strain that only had 1 level of resistance actually showed a similar pattern, while the one that was not that was already resistant to many other chemicals didn't really care. So there's a correlation here between how susceptible you are as an individual and when you give them a choice between sprayed and unsprayed surfaces, where they lay their eggs. And this is precisely why coverage is so important. That if we don't get good coverage, we are essentially giving the target pest a choice between treated and untreated surfaces. And that's what we want to avoid. If they have a choice, they will outsmart us, and they will preferably lay their eggs where their offspring has a higher likelihood of surviving. On the previous slide, I showed you how females shy away from treated surfaces. What we also did in the same study was we used something called EthoVision, which is a behavioral video software program that can be used to measure and quantify movement of individuals. And we looked at diamondback moth larvae, how they also shied away from treated surfaces. The idea is that you follow a larva that you put in the middle of a leaf, and you can see the red track on the left there, how it was actually moving around. So half of the leaf, using the midrib as the separation line, half of the leaf had been treated with an insecticide, spinodoram or gamma cyhalothrin, and the other had been treated with water. And we used sunscreen as a perimeter barrier to keep the larva inside the arena. And then we measured how much time each of the larvae stayed in those 2 zones, whether they was the treated or the untreated zone. And if they were equal amount of time in the 2 zones, we would have what is illustrated with the horizontal line there would be a 50%. But what you can see is that for both chemicals, the diamondback moth strain that was most susceptible to both chemicals actually only about 20 or 30% of the time was spent in the treated surface. So it spent disproportionately, they spent disproportionately more time in the untreated surfaces. So clearly showing that the larvae also shied away from the chemicals. So we basically illustrated that both larvae and adults are able to assess whether this leaf surface is treated with a chemical. And that's why it's so important to have good coverage when these pesticides are being sprayed, because otherwise we're giving the insects an opportunity to shy away. They will find out where to go in order to avoid the chemicals. And this is called behavioral avoidance or behavioral resistance. Returning to our field situation where we want to use pesticides and spray them with the highest likelihood of getting good spray coverage, because we've now demonstrated why that is so important. Getting canopy penetration, meaning getting the leaves far into the canopy covered, and also having good coverage consistently within that canopy. Well, we are the graph as the figure to the right. I think it represents what most people and many people think, and that is the more water we spray, the more water we use, the better coverage and better penetration we're going to get. So it's basically a linear relationship. What I'm going to show you and what we have found in our studies of this is that is definitely not the case. And what we used in this study, which was a very, very large field study, is we used what's called water sensitive spray cards. And these are these yellow cards you can see on the photo to the left. They're yellow. And then when water droplets are sprayed or deposited onto the cards, they turn blue. So you basically have this relationship between blue and yellow. And you can calculate based on coverage if you can find out how much blue you have out of all the pixels, and you can use that as an indicator of spray coverage. If you, like me, enjoy being out and around growers and in sort of real world applications, a good way to demonstrate the potency, the importance of spray coverage, and how likely it is not to get a good spray coverage is by running a simple trial here. And I am standing over here in the middle and showing to some farmers in Western Australia. We created a little field, and then we had it sprayed with a small spray rig that they had, and we placed the water sensitive spray cards at different vertical positions within the canopy. So this would be the top, this would be the middle, and this would be the bottom. And this was with a different nozzle. So in a very simple way, and images speak 1,000 words, right? So you can see this overall with this particular nozzle, the one to the right, we got very good coverage in the top of the canopy. But as we move down into the canopy, we can see we got very low coverage. And of course, when we are targeting pests with an pesticide application, they're not sitting on the top waiting to get covered or soaked in pesticide. This is not how it works. They're sitting inside the canopy, deep into it, and typically on the abaxial, on the underside of leaves, right? So this is really the coverage that we want, that we are hoping will get the job done. So by showing growers this, we are able to then convince them about the importance of this and how proper use of nozzles and how to calibrate your equipment, and also spray at the right time of the day, maybe even spraying at night, is something that they need to look into. In collaboration with strawberry growers at multiple locations in California, we set up these trials. We used their commercial spray rigs. We didn't spray chemicals. We didn't put adjuvants in because we didn't want to include that. That would have made it too big a study. But we did spray at over 3 years in multiple different locations, different varieties with different canopy sizes and different spray rigs, different speed. And because we did it, we sprayed at different times of the year. We had different weather conditions. So we had a weather station here collecting temperature, relative humidity, wind speed, and barometric pressure. during all of these sprays. So water sensitive spray carts had been placed out in these fields, and we then tested different nozzles and could drive the spray rig at different speeds, and therefore get lots and lots of data. In total, we collected data from over 4,700 water sensitive spray carts. So we had, for each of those time points that those cards were sprayed, we had weather data. We knew the volume of water that was sprayed with that nozzle. We knew the tractor speed. We knew the spacing between the nozzles and how many nozzles there were across a bed, because the beds of beds in strawberry fields vary in California. And then we also knew the plant height, the spacing, and the width of the plant. So that had to do with sort of the texture and the density of the canopy that we were spraying. So we basically had three categories of variables. We had weather data, we had operational data, and we had plant characteristics data. And we used those three groups of variables to then run multi-regression analysis and see if we could use them to explain the spray coverage that we obtained on the different spray carts with the different nozzles. You may recall that a couple of slides ago I showed you a linear correlation between spray volume and spray coverage. And you can see why we're a little bit concerned about people thinking that, because this is of course looking more like this is actual data with commercial spray rigs, and it clearly shows you that the story is far more complex than that. That yes, there is a significant regression line when you have 4,700 points, yes, but that line actually only explains about 10% of the variance, meaning that spray coverage, even though spray volume in itself is important, there are many other variables that are interfering here. So we need much more information, and that's why we collected operational settings in addition to spray volume. We collect weather data and also plant characteristics to get much more nuance, much more detail about what are actually the variables that are driving spray coverage. So as indicated with this little cartoon here, this guy clearly has the wrong tool, right? We need better tools to predict spray applications, what kind of coverage we're getting. Because if we don't have that, if there's a high chance of incomplete and poor spray coverage, well, there's a big chance that growers are going to use more pesticides. There's a higher risk of environmental contaminations. They're going to spend more time and more money and more fuel and compacting soil more, because they're going to be driving more around spraying these chemicals. And there's a higher risk of developing both behavioral but also physiological resistance. So we really need much better tools. So that's why all this data that we collected, we have now taken that and developed it into, so the models, the regression models that we then developed, we have programmed them and put them into a phone app. Now, all of this talk about pesticide applications and behavioral resistance and all this was really my own segue to get to where I really wanted to get to, and that is this slide here. Because we wanted to come up with a solution that would be useful. We wanted to help growers solve this challenge of being able to spray their pesticides more smartly. But I am not a programmer. I am there. So I needed help somewhere. So I reached out to undergraduate computer science students, and two of them are sitting here with me. That's Alex and Gabriel. And they, at least back when we started, knew absolutely nothing about agriculture and never thought that their interest and knowledge about phone app development and programming could in any way be relevant to agriculture. But it turns out they could. Once I explained them the project, they got really excited, and Alex was in charge of developing the Android version and Gabriel the iPhone version. So they were working together but on two different operating systems and developing the same program. So we got them involved, and therefore now are able to offer this phone app free to everybody who wants to use it. It's available through my website, and they learned something in the process, and we were able to deliver something that otherwise was impossible because we broke down the barriers between different colleges and departments, and were able to get these very bright undergraduate students involved. We also did something that I think is interesting, and something you should consider yourself. Is we got in touch with a program, a website in England called Futurum Careers. And this is an opportunity to, their goal in life is to encourage high school and undergraduates, high school students especially, to get into science. So we wrote an article about computer science undergrad students and how they got involved in agriculture. So there's an outlet here that really helps promote them and also illustrate this interesting multidisciplinary research program we were able to put together, where computer science and bioengineering and hopes to develop tools so that we can use pesticides more smartly all came together in one project. If you're interested in actually using the phone app either as an Android or iPhone application, or just read more about it, we have tutorial videos on my website in the section called Smart Spray. And you can also, and then you can try it out for free and recommend it to others if you think it's useful. What we really illustrate with this phone app is that it's not only what you spray, but it's particularly how you spray it and how you can optimize that. And we think that is critically important in the responsible and correct way of using pesticides. I also want to point out that I think this is an example of us trying to go beyond publication, because of course we're going to write a publication about that. We have already published similar papers, but it's really about developing the tool that farmers can use. And there's even the possibility that somebody would take this technology and market it in some way, engage with pesticide companies and others and use this. So we really created a platform that goes far beyond the research publication. I can use the app, and I do use it in my teaching, in my lab sessions. So also now I have a gadget, a tool that I can use and therefore enrich my educational program with this. So the app itself serves many important it checks many boxes. But ultimately, I think the most important part is that we're trying to give back to community what we think is so important, and that is that the science we're doing this is leading to solutions that they can use, and the students we engage with learn skill sets that are relevant to the real to the job market out there. In this final segment of this lecture, I'm going to talk about insects as bioconverters of mainly agricultural waste, but really of waste in general. And I'm going to rock the boat, hopefully, a little bit by inspiring you to think about how we may fundamentally be able to change the concept of waste. That waste is something we consider, those are the products, the byproducts that no longer have a value. I think in the future, we have to completely abandon that concept, that everything has a value, everything can be recycled, upcycled, can be used in some way. So we really have to come up with ways of using the waste products, the byproducts from one production system as inputs in another. And we think that insects can play a key role in those concepts. And we truly believe that there's a lot of entrepreneurship to be made, and there's a lot of new entomological research to be done that has to do with within the concept of entomophagy, so using insects as sources of food and feed, but also as insects can be reared to produce oils, fats, pharmaceutical products, many other ways. So they can essentially be small machines, just like enzymes that we can use, we can tailor to have very specific functions in our societies. And we see that as an extremely interesting and innovative way of looking at entomology in the future. Not only do we believe that, not only do we believe, and many other companies are actually developing business models around this, but we all believe that there's a significant potential in this, that waste from agricultural production systems, such as wine waste and tomato peels and skins and other things, hulls from pistachios and almonds, all these that are typically being put into landfills and causing pollution and production of gases and other things. We can actually convert that into biomass, and then we can harvest that and use it to produce fats, meals that can be added to feed, and also the frass, the leftover from the insects in their production. And that can even be taken further and then be used as input for production of mushrooms. And we are looking into that as well. I won't talk about that today, but that's even taking it a step further. So in essence, insects are small machines that we can potentially genetically modify, or we can look at the diversity of insects that are out there that are uniquely specialized to live in all these extreme habitats. Well, a food waste is also an extreme habitat. So what is it going to take for us to associate our knowledge about biodiversity of insects and tie it to find out how we can identify these insects and mass rear them and put them into systems so that they align, so we can align them with the waste streams that we have and turn it into money and sustainable food productions, which I think is a very positive message in a world where where young people are really looking at us old guys, old people, and thinking, what the hell are you doing? What are you giving us? How are we get the cards they are being given are really sort of stacking up against them. Let's give them something positive. Let's look at this as opportunities to create wonderful futures and have meaningful lives based on knowledge about insects and how we can align them with waste streams. There are many very important reasons for looking very carefully into use of insects as a source of protein, fat, and other nutrients. And that is, of course, that their conversion rate, their ability to convert food into biomass is much, much higher than vertebrates. They use much less water. It's much easier to do on smaller scale, so it can be done on rooftops or in smaller places where space is an issue. There's less waste in the process, and you can grind them up, you can store them, you can process it in much easier than you typically can with say meat from chicken or other of the livestock. So really what we're trying to do here is we want to look at insects as a new livestock. We want to look at it as a way, how can we mass rear this and turn it into a serious alternative to the existing livestock production systems that are out there that we know are very, very huge contributors to climate change. As an example of extreme adaptation or adaptability by insects, there are published records out there showing that mealworms can actually consume styrofoam. So we have set up a whole bunch of studies where we put individual mealworms into small vials and we offer them different food sources. This is styrofoam by itself mixed with oatmeal or oatmeal by itself. And then we look at how much we follow over time, and we measure out how much are they actually consuming. And we are applying fundamental principles taken from the livestock industry to see how much variability there is within a population of mealworms, and see which ones are the super performers, which are the ones that really have a high potential, and can we then breed them and create populations from them that will then ultimately become better than where we started out. So basically applying the concept of livestock breeding to insects. And here are some results from that. So basically what you have on the x-axis to the right, you have the individuals ranked from high to low performers. And you can see that in these 3 different populations, so these are different commercial strains that we obtained, there's some variation. And of course, the slope of this line between the highest, the best performers, and the lowest is an indicator of how much variation there is within the population. And we can use this kind of approach to then breed the ones that are the best for that particular source of waste material. And then we could ultimately end up with multiple different strains that have been adapted to different food strains. And these could be licensed by the universities and used by companies to then solve waste stream problems within the ag industry, or in this case with styrofoam, help other managers of waste to turn the waste into insect mass that can then be used for something else. In my lab at UC Davis, and actually across the entire campus, Trevor Fowles is a student of mine. He's in my lab. I'm his main supervisor, but he's actually developing, not only is he a PhD student, but he's also the CEO of a startup company. So he has taken this insect mass rearing and using insects as bioconverters of agricultural waste to the next level. We are indeed publishing scientific papers about that, fine. But he's actually going much further, much further. So together with some of his friends and colleagues, they have started a company called Biomilitis, and they are now offering this. They are working mainly with black soldier flies, but really this is an example of how we are doing some research, and we are trying to then commercialize it through entrepreneurship and also providing insects to people who work in food science and in bioag engineering, and the company is actually selling insects to other research groups around the world. But I really think this is important to think about how the research we are doing, how we can take it to the next level. How can we turn it into what ag should be in the 21st century? Not just about producing crops and livestock, but maybe using insects and using them for production of pharmaceutical products or pet food or even animal food, right? Think of energy drinks or energy bars containing supplements that have been derived from insects, and we could all be part of that, and our students could as well. And how we can make that a positive message in a world where there's lots going on and where a lot of challenges have to be met. With this presentation, I hope I have given you some perspectives on how you might fundamentally change your view on what entomology can and should be in the future, how you can collaborate with engineers, programmers, how you can look at mass rearing as an opportunity for your students to get into entrepreneurship, and in many ways look at how you and your students can meet some of the dire demands by society, how we can empower students, how we can develop solutions, how we can continue to be meaningful, relevant, and really provide solutions and opportunities in the future. Lastly, I want to thank the organizers of this conference, in particularly Felipe, for inviting me. And I'm not exactly sure how the Q&A, the question answer session is going to run here. So if that doesn't work out, if you have any questions or any comments, you're more than welcome to contact me via email. And you can also read more about the research we do on my website. So thank you so much, and enjoy the rest of the conference.